Yeah, I'm going to start out this first session on functional gastrointestinal disorders with a bird's eye view of the concepts of brain-gut microbiome interactions, which has seen a lot of progress in the last few years, certainly compared to the early days when some of us started to work in these areas. And I just want to give you an overview, starting out with where we came from in our concepts of conceptualizing functional or brain-gut disorders to the future. I'll start out with saying a few words about the brain, the gut, the microbiome, and then how these three components interact. And throughout this talk and at the end in particular, emphasize how this model of these bidirectional interactions are relevant for designing effective therapies. So the traditional concept of IBS, which have been around certainly as long as I've been at UCLA, have been that there's a camp of people that think this is a psychological problem related to anxiety, stress, and trauma, and then another group, the larger group, that over the years has sort of favored different hypotheses from motility disturbances, secretory disturbances, bacterial overgrowth, and food intolerances. For me, who's been in this business long enough, it's kind of amazing how every 5 to 7 years the emphasis on these topics has shifted. And I think we will see or at the end of this talk, you'll see a significant change in this. So one thing that we got interested early on was what role the brain plays in this disease. So away from the psychological viewpoint to a brain science viewpoint. And over the years, I've really done a lot of work to, I think it's actually not yeah, I've done a lot of work to corroborate this concept, and as you'll see, it's made significant progress. So, in addition to the brain science and the science about the gut connectome, all the cells in the gut that interact with each other, there's also been a shift away, a gradual shift away from believing that simple single targets, selective targets in the brain-gut axis are responsible for a complex syndrome like that, but it's really the bidirectional interaction between the networks in the brain and the networks within the GI tract. So let me say a few things about the brain, and this has sort of really made a tremendous progress over the years. So using techniques that are sort of summarized under brain imaging techniques, there's a whole range of those, including functional MRI, but also structural and MRI spectroscopy. We have identified that there are certain networks in the brain that are relevant, that are abnormal in IBS patients, both structurally and functionally. One is the sensory motor network, so consistent finding in all studies that there's an increase in the somatosensory cortex in patients, and not just patients with IBS, but also patients with many comorbid disorders, comorbid functional disorders. Probably most importantly, the insight that there is a system in the brain that is activated continuously. It's sort of like in self-driving cars, that the cars gather information and constantly compute the prediction of what's going to happen next second, next 30 minutes. The brain does that for even longer time frames. So this so-called salience network, which assesses the importance, the subjective personal importance of internal and external stimuli, makes predictions about the future, and then whenever this network perceives something as important, a potential threat, it will engage attentional networks, so the central executive network, and the emotional arousal network. So all these networks that work together in the brain, we've been able to dissect and identify abnormalities, specific abnormalities in the IBS patients. The last consequence of this process is, and this all happens obviously within milliseconds, that there's this engagement of the central autonomic network, that network of the brain that sends signals back to the body in terms of vagal, sympathetic signals which regulate motility, secretion, gut permeability, immune function, and the microbiota. So from the beginning, when the brain assesses what's important, what's threatening, all the way to the output to the gut, we have been able to identify this. I should also say, unfortunately I didn't get the updated version of my talk here. I had a picture of Mellinkoff on it because when I was a junior faculty member, it was really exclusively because of Sherm that we got started in this area of brain science and functional GI disorders, because nobody at UCLA was willing to let us use that scanner. They thought gastroenterologists have no role to play in something as sophisticated as brain imaging. And it was only his intervention that we got the first 5 pilot scans that allowed us to start this research. Now, these concepts have a lot of implications for therapy. So cognitive behavioral therapy, minimal contact cognitive behavioral therapy, a 4-hour session with patients has been shown, and there will be a big study coming out in the near future, to be highly effective of treating IBS symptoms. And so people often wonder why cognitive behavioral therapy is just another psychological intervention. Well, it's actually not. It's really a biological therapy that changes the gain of the salient system, both in terms of its structure and in terms of its function. So if you send a patient to undergo this therapy, it will change the brain of people. In terms of the attentional networks, so CBT, hypnosis, mindfulness-based stress reduction, all these interventions, if you look at it, actually have better NNT numbers than most available medications. Emotional arousal network, similar relaxation, abdominal breathing, stress reduction. And then finally, when we go to the central autonomic nervous system, we haven't really figured out how to target this in the brain pharmacologically, but most of the therapies that are aimed at the gut to deal with secretion, up or down regulate secretion, or motility, really deal with the aberrant signals that the gut gets from the autonomic nervous system. So we have therapies, some more successful than others, and some of the novel drug therapies, most, in my opinion, fall in the last category. So a few words about the gut. I mentioned this term gut connectome before. So there's been tremendous progress in identifying multiple cell types in the gut, shown here, and will not go into the details of this. So there's the largest portion of the body's immune system, gut-associated immune system, the largest portion of the endocrine system, all these enteroendocrine cells. Almost entirely the serotonin content of our body is in the gut, more than 95%. The biggest nervous system outside the gut. So a lot of very important regulatory systems placed in the gut. If you spread out, it's about the size of a basketball court. And the latest addition to this complex system is the microbiome. And what we know now from many studies that all these cells can communicate with each other. So you can't really identify what one cell or one microbe or one neuron does or one receptor on those cells. It's really the interaction of all these cells together. Now there has been a tremendous effort in the last 30 years or so, and we have been involved in this with different companies to identify targets in this complex gut system, and a variety of compounds have been proposed that with benefits, often tremendous benefits in mouse models or rat models where this has been tested, just listing some here. The sad thing is the great majority of these has not translated into effective therapies in humans. So even if we thought that some of these things have to work in humans as well as in the mouse, it did not happen. So the gut microbiome, the newcomer to this concept of the brain-gut microbiome axis, just a few things. I mean, this has almost become common knowledge now because it's in all the, in the lay press, it's been featured and digested many times. So just a few words about this. So we have as many microbial cells as a human being as we have human cells, and more importantly, the genes that these microbes have and which are constantly evolving, there's not stable gene pool like our humans, it's been estimated to be 400-fold greater than our 20,000 genes. So the potential capacity of these organisms, we're just scratching at the surface. Based on this vast gene pool, it's been estimated that these microbes can produce up to 500,000 different molecules with which they talk to each other, but also can talk to us, to our GI tract and all the organs in our body. Even though invisible, the microbiome has been estimated to be about the size of the liver. So just imagine if textbooks have been written in gastroenterology without the liver. If you hadn't known about the liver, you would think they're completely useless. Well, all our textbooks have been written without taking this additional organ into account. So there will be a lot of surprises in the next few years when we realize what this system does. And one last thing which is kind of interesting, so based on our human genes, we're nearly identical to each other. But if you look at the people sitting around you, you share you share very little of your gene pool with your neighbors. There's many influences on the gut microbial composition and function. So the composition we usually refer to as the microbiota. The function includes the genes and the activities that they can produce, and that's called the microbiome. You see from diet, genetic background, interaction with the immune system, antibiotics. All of these play a very important role, both early in life and during adulthood. But interesting, and we've sort of been used to this as people that are looking at the brain as well. The publications, most publications do not include the brain. This is a particularly intriguing topic. There's only a few research groups working on this, how our complex brain can communicate with these. And so we got interested in this and fascinated by, after Kirsten Tillich in our division did sort of a seminal study, which remains really the only evidence in humans for this interaction, where she took healthy young women. These subjects went on a 4-week course of daily ingestion of a probiotic-enriched yogurt. And then she tested them with the brain imaging techniques in response to an emotional stimulus. And make a long story short, a complex study, what happened at the brain network that was engaged with this emotion recognition task, the connections in this network decreased with the probiotic intake, meaning a less robust response to this emotional stimulus in the women that had taken the probiotic. In the meantime, a complex interaction between the gut microbiota, the peripheral immune system, and the brain has been identified. Won't have time to get into the details of this, but just some of the molecules or signaling molecules, pathways that play a role, are neuronal pathways, the vagus nerve particularly, neuroactive molecules that the microbes make, such as neurotransmitters, serotonin, GABA, and many others. And there are many other microbe-derived molecules, such as short-chain fatty acids, which target many of these cells in the gut, and through that can influence signaling to the brain. So for example, with satiety, many of the endocrine cells that contain the incretins in our GI tract, like GLP-1, also have short-chain fatty acid receptors on their surface. So the microbes have a big influence on setting the gain of these cells that then send signals to the brain. This complex system is programmed, and this is one of the key points early on in life, both during pregnancy, but also during the first 3 years of life when the microbes are being established. Once it's established, it's fairly stable and robust throughout our lifetime. And then the only thing that happens throughout life without major changes, unless you develop C. diff, which is one of the few exceptions where you knock out this complex system. Diet plays a big role in modulating the basic tone of the signaling from the microbes to the rest of the body, including the brain. There's also an important thing, so you would think that the brain is sort of separated from the gut by barriers. So there's a lot of talk about the intestinal permeability. Leaky gut has become sort of a common household word that all of our patients come and say, I have a leaky gut. Does that explain my IBS? We know a lot about this scientifically, shown on the lower slide. Multiple pathways. The mucus layer plays a big role in determining the gut permeability. If you're on a high-fat diet, the thickness of the mucus layer will decrease and the permeability will increase, so more microbial substances can get into the get close to the gut's immune system. And on the left side, multiple pathways have been found. The actual epithelium can become leaky and let things through. The factors that play a role in this are stress, inflammation, immune activation, gut microbes, and diet. So all these factors determine how much of the information that the microbes have in your gut makes it to the brain. And at the brain level, so this is blood vessels, blood vessel within the brain. Similar factors can modify this blood-brain barrier permeability, stress, immune activation, and microbial mediators. So a highly flexible system of how much information can get from the gut to the brain, with immune activation and stress being sort of the two major factors. Now, we talked a lot about how the brain can talk, how the microbes can talk to the brain. It goes the other way as well, and this may be equally, if not more important. So there's stress-induced changes of the microbial environment or the context. From many studies, we know that stress can change regional GI motility, slowing it down or accelerating it, fluid secretion, mucus secretion, and many other things that I'm listing here. And then, possibly most interesting, the sympathetic nervous system can directly talk to your microbes as well. So when you're stressed, norepinephrine not just drives up your heart rate, but it leaks into the gut and changes the behavior of the microbes. There's a good example, for example, with post-infectious IBS. Significant stress, psychosocial stress around the time of the infection, the initial gastroenteritis, plays a big role in determining the duration of the symptoms and also the development, likelihood to development. In my opinion, a big factor is this modulation of your gut microbiome by stress, which then creates a different response to a pathogen and to the microbial host interactions. So we put this all together. We have a very complicated system, 100 billion neurons, 85 billion glial cells in the brain, so the immune cells, 100 million neurons in the gut, and I mentioned that earlier, the components. And so when we talk about stress, people always say, yeah, every disease is stress-related. This is something non-specific. Well, if you look at this system as a bidirectional brain-gut interaction, there's two types of stress. One is psychosocial stress, which always not just activates the brain, but always has a reflection at the gut side. But there's also gut stress, such as diet, high-fat, high-sugar diet, that is characteristic of the North American diet, antibiotics and infections. And similar to what happens with brain stressors, any change in that gut connectome will always have a reflection at the brain level, no matter if you feel it consciously or not. So I've worked in this area, and several of us, Lin Chang, has been part of the center from the beginning. So we've always been interested in brain-gut interactions. Nobody was really interested, neither our colleagues in gastroenterology nor anybody outside. So in the meantime, it's been an explosion of putative brain-gut microbiome disorders, and I make a long list here, and just want to say a few things about IBS. And there's different paths that sort of going on in investigation. So one is there an altered gut microbiome in IBS patients? This is a short selection from a long list of publications on this topic. And it's difficult to get any sort of consistent message out of this. The latest conceptualization is that there's not one microbiome or one IBS alteration or dysbiosis, but there's several subgroups, and Jeffrey was the first one that published this. In the meantime, we've also done a study which basically identified these are a representation of the main taxa of microbiota in IBS, healthy control on the right side, and then two groups of IBS patients, one indistinguishable from healthy controls and one with a different composition. And then we found, and again, complex story, but simple conclusion, that if you compare the two IBS subgroups, the one with the normal and the one with the abnormal microbiota, there were differences in brain structure. So there was a correspondent image to the microbial dysbiosis to brain alterations. So for example, the abnormal IBS group had smaller volumes in prefrontal cortex and the insula, and larger volumes in the sensory motor network region. Remember what I said earlier, the sensory motor network can become the footprint of functional GI disorders. So this may be one of the reasons. I'm not going to get into the basic sciences. I just mentioned this. So a study recently came out from the group at McMaster, which characterized, again, the microbiota in patients, selected group of IBS patients with and without anxiety. And then they took the fecal samples from these individual patients and transplanted them into germ-free mice. And what they found, primarily the subgroup of IBS patients with high anxiety, showed difference. The mice that received that transplant showed differences in behavior and in bowel movements. So something is in the stool of the IBS patients with anxiety primarily that can induce changes, kind of parallel changes in the mouse model. So synthesizing the information that we have currently from a series of data, I'd like to propose this following model. Clearly there's genetic predisposition and epigenetic alterations occur early in life in individuals that are more prone to develop IBS throughout life. Then there's trigger factors, psychological stress, like could be chronic stress or trauma. But alternatively, they can also be, this whole system can start with gut stress. I told you there's a complete integration of the two systems. It can start on one side or the other. Antibiotics, dysbiosis, infections, all this can happen early in life. The result is there is stress-induced alterations of the gut connectome, dysbiosis, increased permeability, leakiness, and immune activation. This will generate, as I showed you earlier, signaling molecules from the gut microbes that can signal back to the brain, either in an endocrine fashion or through the vagus nerve. And then ultimately, these signals can alter brain structure and brain function. And when we see an adult IBS patient, most likely, no matter where we look at the brain or at the gut level, we'll see these alterations. Depending where it's started, and also depending on the predominant symptoms, different kinds of therapies will be more effective than others. And another conclusion is that single targets will most likely have small effects in a complex system like that. And that's exactly what we observe. So the therapeutic implications, in terms of psychological stress, to come back to what we talked about earlier, so stress management, cognitive behavioral therapy, At the gut level, dietary interventions, pre and probiotics, term of psychobiotics, really established just in mouse models, very little evidence yet in humans that actually does occur, that you can take a probiotic and affect your mind in a significant way. And modulators of secretion and motility, so the drugs that we have today. In terms of the alteration in gut-brain signaling, that's really more future. A lot of companies, startups are working in this area, identify analogs of microbial metabolites that play a role in this feedback to the brain, and modulators of microbe-gut interactions that play a role in generating some of these molecules. And then at the brain level, again, something that's currently being studied with great interest and great investment, identify analogs of microbial metabolites and modulators of microbe-gut interactions. So in conclusion, I don't know how many people I convinced in this room of this concept. So IBS is a disorder of altered brain-gut microbiome interactions. As Lin Cheng will emphasize, there's even discussions in the Rome process to rename functional disorders of brain-gut interactions. Dysregulations, and I think this is an important one, of the system may be initiated in the brain or in the gut in different patient populations, and this may have implications for the optimal treatment. Treatment aimed at single targets within the brain-gut microbiome axis have small effect sizes and characterized by high NNT numbers, and we have certainly experienced this over the last 40 years with many of the developments of novel drugs or attempts to develop novel drugs. Current treatment strategies aimed at the gut microbiome have small but significant effects in terms of probiotics. They're not dramatic, but and they differ between different patients. Some are often temporary, like the FODMAP diet or antibiotics, and have uncertain long-term effects on gut microbiome. On the gut microbiome, particularly antibiotics. I think, in my personal opinion, in this day and age, to give a broad-spectrum antibiotic for a relatively benign disease, particularly if you give it repeatedly, all the stuff is coming out about the importance of the microbiome, I don't think is a wise approach. And optimal treatment strategies, in our opinion, should be aimed at multiple sites within the brain-gut axis at the same time, including the brain microbiome-related targets and mechanisms, diet and lifestyle. So a general multidisciplinary wellness approach is almost certainly work better than any sort of single therapeutic strategy. So thank you very much for your attention. If you want to read more about this, get my book. Thank you.